Welcome back to episode 25 of Michigan Brews. I think I lost your audio, Brian.
Oh, really?
There it's coming back. Yeah,
all right.
It's just quiet. So as you see, Jordan's not with us tonight. I think he's off playing dad again. We're not feeling quite up to it. So it's just going to be Brian and I. We've got a cool guest tonight, though. Really happy to have actually Dr. Lance Shaner, Esquire. I want to make sure I got that right, from Omega Yeast, the founder and co-founder, co-owner of Omega Yeast. So he's going to get on and dig into some yeast facts with Brian as I sit here and get drunk. So that'll be it. So welcome, Lance. Thanks for joining us.
Thank you.
I got the title right?
Yeah. Well, I guess one thing about PhDs, though, most of them don't require that they're called doctor.
Okay.
You technically could, but the Esquire is a nice touch too.
I was actually digging through old, articles and interviews, and I saw it referenced a few times, and I just thought I'd go with it. So
yeah.
I didn't put it in the show description, and I didn't know if I'd piss you off.
No. Yeah, I've got enough letters at the end of my name. Yeah.
So we usually start off the show talking about what we're drinking. Sometimes it's homebrew. Usually we have a brewery on that'll tell us, or we're drinking some of their stuff. But since you kind of supply all breweries, or a wide variety of breweries, we didn't get anything specific to you. But what are you drinking tonight? What are you having?
Revolution Riot.
Nice.
A Bohemian Keller Pils. It's an unfiltered pills. It's very good.
Oh, is it with your yeast?
I don't know. I don't think so. I don't think they've gotten live, but they were picking up yeast and dropped off a case today.
Oh, nice.
Which is one of the perks of the business.
Yeah, no kidding. Was it all that the Keller beer? The Keller, what you call it? Keller Pils?
Keller Pils. I don't know. I was just handed a 4-pack from what they brought. Whoever got it knows my predilections.
Nice.
Yeah,
I don't have much Revolution up here.
No, very little, I think.
Mostly just like their IPA, like mixed packs and stuff like that.
Yep.
The Anti-Hero and stuff.
Yeah, as much as I think they do try to keep it, pretty Chicago centric. But they make a ton of beer, so it's actually surprising they don't have a larger footprint. But they hit it pretty hard around Chicago.
Yeah. I think it started coming up last year, but yeah, it's just like the Anti-Hero and the Hero and the some of that. It's not too much. But what are you drinking, Brian?
So I'm just finishing off a beer and probably going to crack another one in a minute here. So on Black Friday, new brewery in Grand Rapids, Michigan, just launched their first beer, Black Calder Brewing. It's the first fully black-owned brewery in Michigan, I believe. They made a black IPA with Mosaic and Idaho 7. So that's what's left of this guy here. But it's delicious. I mean, it's like, you don't see black IPAs that often, right? Anywhere, or at least not these days. I guess there's a time period where they were all over the place, probably. But I don't know. It's as good as any I've ever had, if not, the top of the top. I love Mosaic and everything anyway. So
nice.
But
actually, it's funny. It's the only beer I have tapped in my new kegerator is a black IPA.
It's a black IPA,
and I'm not even drinking it tonight. So. I'm pounding some seltzer, dude. Lance, with proper seltzer, I think you're going to change my game. I've got one going in the basement right now, and I was going to talk about this later, but what the hell? I picked up Lutra and proper seltzer last week as soon as our homebrew store got it, and started kicking off a seltzer. And I'll say it's not this, but it's the cleanest, clearest, nicest seltzer I've ever made, or even a homebrew seltzer. It's like usually I hate my shit because
always the harshest judge.
But go ahead.
Oh, that looked really clear, like in terms of color and stuff too.
Oh, this one, this is Offside's cucumber.
Oh, okay.
Yeah, no, the one I made with Omega's Lutra and proper starter. Probably crash it later this week. My basement's a little cooler, so it's fermenting slower. It's about 7 days in. It's gone from 1080 to 1028.
1080.
I did.
Yeah, are you gonna
I'm
water
sorry.
that down?
1070 to 1030, 1028, and my intent was to brew it a little high and then cut it into 2 batches. So,
okay. And actually, if we want to jump right into science, there's one of the interesting things we did is fermented high gravity seltzer like that, and then cut it. Well, we sent it off for analysis for the fusels and esters and all that. One of the interesting things when you compare, say, a 10 Plato starting seltzer to a 20 Plato, you don't get 2 times the amount of esters or fusels in the 20 Plato. So effectively, then when you cut it back to say like what would have been a 10 Plato start, it tastes cleaner. So because you're not doubling all of those other things. So it's actually a really, really good technique for getting a very clean seltzer base is to brew it at higher gravity and cut it back.
So I and I miscounted on the bags of rice syrup solids that I grabbed. I was doing a mix of dextro and rice syrup solids. And so when I got home, I thought I had 4 and I'd only grabbed 2. And so I hit 1070. And I told my wife I was a little bummed because I did want to brew that higher gravity and then, split it into like 2 kegs that I could flavor differently, cut it with some water. And so anyway, I didn't grab enough sugar. I didn't get there. I got 1070, but here I am a week later. It's 1028 right now. I pulled a sample today and we both drank the sample and it's like, this just tastes like really clean, slightly sweet water. Even at that point, it's probably around the 5% ABV mark. And she compared it to just a really delicate wine, sweet wine or whatever. And I was just, I even texted Brian. I'm like, dude, this shit, I could crash it right now and carbonate it and not even flavor it. It tastes just clean, slightly sweet, and really refreshing. So
yeah, to me, the unflavored ones always taste like sake, just like a very sake-like, which I think makes some sense because sake is essentially just rice is relatively flavorless, so it ends up mostly being just dextrose when it's broken down. So it's just that's what it tastes like. Yeah, it always tastes like sake to me, and it's just very easily covered up with whatever flavoring you want to do if you don't happen to like sake.
Yeah, no, but anyway, So now, normally Brian and I are really good about going off topic. Not so much about jumping way the hell ahead to when I was going to start talking about seltzers. So sorry about that. So
like the seltzers are like it's still like I thought it was going to be like a passing sort of like thing like the black IPAs, like, oh, it's going to stick around for a little bit, and then like never really got into them. And then we had our like seltzer episode not too long ago, and they're obviously still on the shelves and stuff. And Lance, you guys are focusing a lot on doing stuff for seltzers and with the proper seltzer thing. And I'm gonna make my own first seltzer soon because they're not filling. I think that's my thing with like drinking a lot. Sometimes it's just like, man, I'm like full. Like I could still go to for another couple beers or something like that. But
yeah, and I mean, and they're just like a blank canvas too. I mean, you can kind of get creative with your flavoring. So it's just some place where you can be creative.
Yeah, I mean, and we did that seltzer episode, and we lined up what was it, like 8 different seltzers. And normally, like when we do a style show, so we do a few different kinds of shows. We'll do a show where we'll talk about a style. We'll do a show with a brewer or guests like you, and then sometimes we'll do shows with, home brewers or clubs or things like that. And so we had done, Brian and I don't think Jordan was there, but Brian and I did the seltzer episode, lined up like 8 cans of seltzer, and before you know it, the night had gone horribly wrong. Because they were just going down so easy,
5% at a time.
But yeah, no, I'm super excited about proper seltzer and the Lutra combination. I really dug it. It fermented clean. The previous batches of seltzer I've done with either champagne yeast or just regular yeast that I tried to kind of treat with my own DAP or whatever. They always had a winey characteristic or just kind of this off flavor that I didn't like. and this one is super clean. So
yeah, and the wine yeasts are always very sulfury too, especially if you don't have that nutrient mix, right?
Yeah. So
is there any? Oh, go ahead.
Oh, I was just going to jump back into Omega.
But I was going to ask if there was any other like yeast that Omega had that you would recommend besides, Lutra for seltzers.
Yeah, I mean,
seeing success with.
The one kvike that we haven't is the hothead. It doesn't seem to work that well in a seltzer environment. But we did. I mean, the nutrients tailored really for it to be useful for pretty much any brewing yeast. Okay. So with any brewing yeast, you should be able to ferment it to completion. That's got everything that any ale yeast would need. But we do have a lot of, the kvikes, especially if you push them warmer, they'll chug through it pretty quick.
Cool.
Right on. Yeah. So we should go back to the beginning. Give us your background and how you got into. Why are you a yeast producer?
Yeah, I guess we could step back to college. So where most beer drinking adventures start. So I went to the University of Illinois in Champaign, and when I was like a sophomore, I think, I joined. They have a homebrew club there called Buzz, the Boneyard Union of Zymurological Zealots. And it's an actual university sanctioned club, as in they have a booth on Quad Day out on the Quad. And they're very, very active club. So I don't remember if I think I might have started home. I don't know, this is so long ago now and so many beers ago. I don't remember if I had started to brew first and then found them, or found them and started to brew. But attended those meetings regularly, and of course learned that you could buy everything you needed to brew with before you're 21, because it's just grain and yeast and nothing alcoholic about it. So it was kind of a backdoor to getting beer too. But there were some world-class brewers in that club. So it really opened my eyes to what you could do at home. It's like you don't really need fancy things to make great beer at home. So it was very inspirational as well. So kept brewing. And then there was also somebody in the food science department who had a huge collection of yeast too. So you could get whatever strain you wanted. He'd give you like a little 5 milliliter starter that you could step up at home. So it also gave me my first kind of experience in handling yeast. So from there, ended up moving to Houston, Texas, to go to graduate school. And God, that was even a beer-related venture, because I always tagged along with the graduate students when the department would have speakers from other universities come in and talk about their research. And afterwards, the graduate students would take the speaker to this local beer and bread shop, and we'd eat some good cheese and bread and have beer. And I was tagged along with them, just mostly to get free beer and cheese. And it happened to be around the time I was applying to graduate schools, and somebody from the University of Texas in Houston had spoken there. So I was talking to her, and she's like, "Oh, you should apply at our school. It's a good school." And I'm like, "Oh, I hadn't thought about that. I'd been looking at others, but I hadn't considered them." So I applied and flew down there and really liked it. I liked the people I had met. And she was working on something really cool. She was actually working on Bacillus anthracis, so the bacteria that causes anthrax. So I'm like, well, that's pretty cool. I'd like to work with anthrax. So moved to Houston, and just the funny way things work. The first you do rotations in your first year in graduate school there. So you pick a lab and you're there for like 2 or 3 months. Then you go to another lab and do some projects there, and then another one, and then you pick the one that you like the most, had a good rapport with the professor, to do your full thesis. So the anthrax lab happened to be the most popular one. So there's like 3 students out of the incoming class of 9 that were all wanting to do their first rotation there. I'm like, I don't want to go to the same place where everybody else is going. Right. So I ended up choosing to rotate through Kevin Morano's lab. He's a fairly new professor there, but he was working on yeast. and on Saccharomyces cerevisiae, not in the brewing context. It was more in the human health context. But I'm like, I like to brew. I like to work with yeast. It'd be fun to actually get more hands-on with it. So I joined his lab and just fell in love with yeast. I mean, just working with yeast is so. I mean, they smell good. They smell much better than an anthrax lab for sure. And you can just do pretty much anything with them genetically. They're just so genetically malleable. I mean, you can do neat things like take a human version of a gene and replace the yeast version, and it'll serve that same function in the yeast cell that it does in the human cell. So you can study human conditions using yeast as your model system.
Wow.
So that was the focus of the lab. We studied the stress response, which has a ton of parallels between human cells and yeast cells. But along the way, I got to store my brewing yeast strains and grow them up in whatever quantities I needed to brew. So I was always brewing through graduate school, which makes you popular in graduate school too, to have kegs available all the time. So yeah, I ended up liking it a lot, did a rotation through one other lab, which wasn't as interesting, and then went back to the yeast lab to do my thesis work. So it was just, I mean, I went to Texas thinking I was going to be studying anthrax, and ended up studying yeast, which had obviously a huge impact on what I ultimately did. So it was just, I don't know. It's just funny to think about it in retrospect, and how you ended up where you did. And just these funny little turns. I mean, life might have been different had not everybody wanted to go through that anthrax lab at the same time. But anyway, so from there, once I graduated from there, actually had gotten kind of disillusioned with doing bench work. So I took a detour to law school at that time at the University of Houston. And the whole point, or the whole goal going into it, was that I was going to do patent law when I got out. Because you have to, in order to practice patent law, you have to have a technical degree and the law degree. So basically you act as the go-between between the inventor and the patent office. So you have to understand the technology and the law. So that was the goal going into it, that I didn't end up doing that. So when I graduated, I got a job at Marshall, Gerstein and Boren in the Sears Tower in Chicago, and moved back up to Illinois, and became a patent attorney, and did that for about 4 years. And it was during that fourth year where I had a very chance conversation. Again, another one of those, right moment, right time, where I was talking to Andy Smith, who was one of the founders of 1090 Brewing in Glenview. And he was just, they were still in the planning phases, and he was just talking about where he's going to be getting his yeast. And so we were just talking about yeast, and just occurred to me during that conversation that there's no yeast labs in the Midwest. Nobody's doing this. And it's a perishable product. It has to be shipped quick and cold and all this. So we, in my head, I'm like, we can get off the ground by just focusing on Chicago. There's a lot of breweries in Chicago. It's growing as it is. And so it seemed like it could work, which, in retrospect, it seems ridiculous too. It's like, I don't know. I've never grown yeast on an industrial scale. So I was probably like naive to think I could do it. But, I talked to one of my colleagues at the firm, Mark, who became my business partner, just about the idea, and said, I think this could work. This is what I think the inputs will cost. And again, not really knowing all that would be involved. But he was intrigued enough by the idea. And I was really just talking to him to kind of like just talk it out. But then a few days later, he's like, "Do you want a business partner?" And said, "Sure. I don't know what I'm doing, so let's give it a try." So this would have been like November of 2012, December 2012, when that conversation happened. By March of 2013, we had space rented in a costume shop in Chicago, where they had basically walled off a section, installed some plumbing in it, and we had our 1,300 square foot lab to start out. Wow. And basically just a moonlighted. I mean, we'd go in the evening and kind of do everything we needed to do, get home at 1 a.m., wake up, take the train back to work to be a lawyer for 8 to 10 hours, and then go back to the lab and do that till 1 a.m. And it was exhausting. And got to the point where we actually started to get enough business where it's like, this isn't going to work if one of us doesn't just take the leap and do it full time. So I think August of that first year is when I just informed the firm that I was going to leave and do this full time, and felt pretty good about it. Still, obviously, still feel pretty good about it. But it was, I mean, it was tough that first. I think for the first 2 years, I literally was at the lab every day for 2 straight years. It's in a lot of ways, it's like raising farm animals. I mean, they have to be fed daily. They have to be, cared for. So you really can't take a day off. I mean, you have to be checking on them to make sure, things aren't blowing up. And it was rough until we could start to hire more people and get people that you trusted to do what you would do in the same situation. I mean, there's one Thanksgiving where I had got food poisoning or something, and had to go into lab the next day to do things because there's nobody else to do it. And I could barely walk. That was just from fatigue, but so thankfully those days are long past us, and we have a lot of people that we trust and awesome. To get things done. But it was a struggle for those first couple of years, but that was the beginnings.
It's nice to know that you're kind of the same. I've talked to a lot of brewers that have started their breweries because they were talking to a buddy, having a couple of beers, and said, "This sounds like a crazy idea. Let's do it." And they've made successful breweries. And then here you did it with a yeast, manufacturing company as a whim. So that's kind of cool.
Yeah. I mean, it probably is the same story. It's just the supply side of things. And the analogy that my business partner, I don't know if he made it up, but it's probably relevant in other situations too. But basically, during the gold rush, would you have rather been the miner who had a chance to do really, really well, or would you rather be the person selling the pickaxes?
Did your buddy watch Deadwood?
Did they talk about that in Deadwood?
That's pretty much what the central, one of the many plot lines of Deadwood is the main characters are the hardware store salesman that moved into Gold Rush. Yeah. So. Yeah. Well, he put that into different. Well, yeah, maybe he did. That'd be funny if he, because I watched that series. I should have known he was ripping off Deadwood. But you're right. Like, yeah, why go out and bust your ass to do the work? Sell all the tools to do the work. But this is going back to so you were nice enough to come to our homebrew club meeting last month and do a demonstration at the homebrew level for yeast. And one of the things you said kind of ties into this is, and I'm losing it. You're one of the largest beer producers in your area, right? But you're not actually, you don't sell any beer. You guys brew a crazy ton of beer to make your yeast and grow your yeast, right? Yeah. I mean, and in our old system, we used dry malt extract, and when we were beginner homebrewers, just using extract to grow our yeast. And when we moved into the new building, we actually got a brew house. So we have a 4-vessel, 30-barrel brew house. So we use malt. We have a silo of malt out back. So we are, basically, we're brewers that dump all their beer down the drain and keep the yeast. So we yield like a 4% alcohol beer that's obviously heavily oxidized because we're feeding air to it all the time to encourage yeast growth. But we brew 30 to 60 barrels of beer a day. Yeah.
Wow. Where so going back to like your beginnings and stuff like that, and maybe this, I have no idea how like yeast labs start. Where do you start to like acquire like your strains from and stuff like that? Like, you obviously just don't start and magically like have yeast on hand. Like, how does that work?
Well, I mean, it's, again, since I started brewing in college, I mean, basically when I moved to Texas, they had all those strains in their collection there, so I got streaks out of all those and took them with me to Texas.
Cool. Okay.
And bought yeast over the years that I then stored away. Just, I mean, like I hear you have your own yeast lab. I mean, the thing about these strains is that they're not. I mean, before White Labs was selling Chico, Siebel had it in their collection, and Sierra Nevada was brewing with it. So the strain existed, and there's nothing protecting the strain itself. There is intellectual property that protects what you call it. So I can't sell a strain under somebody else's trade name for the strain, but the strain itself is not protected. So that's so part of it with us is making sure we have all the strains that brewers are used to using and want to use, but also finding and making other strains. So the whole gamut. We're full service. If you like using Chico, we can provide Chico. But if you're looking for new things, we're again trying to find like Kveiks. I mean, we were pushing those before anybody knew what they were. We got those first in 2015. And then hybrids. We were the first to have hybrid yeasts out there that we created ourselves. So yeah, I guess we'd like to say we're full service.
Is that so for the intellectual property there, or what you just talked about? How does that apply to something like your Javaru strain? Like, I mean, that you got from a specific Lithuanian source, right?
Yeah,
and you, that's a proprietary yeast for you. You have an agreement with them, or however. So can anybody come along though and just regrow that yeast and sell it under a different name?
They could, and there are other places that are doing that. Yeah, the Yovaru one is actually kind of.
Sorry.
Sorry. Yeah, no, I called it Jovaru for the longest time until they told me that the J was pronounced like a Y. It's a different one because most of these you can get something out of a bottle, you can sell it under whatever name you want as long as it's not like a brewery name, something like that. With Yovaru, they sent us the strain and we got to use their name. So Yovaru is the name of that brewery. So we filed a trademark application on behalf of Aldona and sell it under her trade name. She owns that trademark. We just filed it for her. So nobody else could sell something they call Yovaru. But yes, there's nothing we can do to stop anybody from doing it. We do also, though, have that arrangement with her where she gets a percentage of our sales. So other people that are selling it don't have that arrangement. Right. And she always was very protective of the strain. I mean, she doesn't want other people to have that, to be selling it without her getting something for it. But there's nothing she can do about it. There's nothing we can do about it, other than to publicize that we do have this arrangement with her. Like we got this strain from her with this agreement for her to get, royalties. So that's the most protection we can get for something like that.
Well, that's interesting. I mean, because I thought the same thing that Brian had is, where do you come up with these strains, or how do you get these strains? And one of the things that I read from you, I think back when you were doing research on the Brett strains, was you were like, the best way to get them is to do dregs, or the best way to get clean Brett strains was to do dregs off European sours.
Yeah, lambics are loaded with lab Brett.
And so I wondered, like, is that a thing out there? Are the yeast companies like yourselves and the other ones out there, are they just getting dregs and samples from other places? Like, how did you come up with all these bikes? And I knew I found the story on the I'm going to screw it up again. Yovaru. Mm-hmm. But how did that turn into, or how did you start getting involved in all of the Vike strains?
So we found out about it probably the way a lot of people did, which is reading Lars' blog. I read that and reached out directly to Lars. I'm like, I got to try these things. They sound incredible. and he didn't actually get back to us right out the gate. I don't know, didn't know who we were or whatever, maybe getting inundated. But so but he had mentioned in his writings that he was, submitting them to the National Yeast, NCYC, National Collection of Yeast Cultures in the UK. So we just reached out to the NCYC. We're like, can we, and you can order yeast from them, and they'll do a licensing deal as well. So that's how we got Hothead, just based on what they had and what Lars's description of it was. And I'm like, this sounds like a neat one to start with. So we sent off for that. That's the Stronda Kvike that we called Hothead. Just because, again, 2015, nobody knew what Kvike was. So if we had come out and sold something as Stronda Kvike, we wouldn't have sold any of it because nobody knew what it was. Now that changed to where we now release things with the Kvike name, because people know what Kvike is and people search for that sort of thing. But at the time, that wasn't the case. So that's where Hothead came from. At some point after that, we had reached out to Lars again, or he finally answered or something. And so we've got everything we've gotten subsequently has come directly from Lars.
Cool.
The other, I don't know, I think the three other, the Hornindal, Voss, Espa, those all came directly from Lars, and a couple others that we brewed with and didn't seem like they necessarily added anything beyond what we had. So those are just sitting in the freezer.
And then the other ones, like and things that you're coming up with.
So Lutra came out of the Hornindal blend. So when we really dove in and picked apart the Hornindal culture, we found 9 distinct strains in it, at least 9 distinct strains, and then brewed with those individually to find Lutra out of that blend. So it is a single strain component of Hornindal.
Is that whole blend what you sell right now? Like all 9 strains are in like that Hothead packet of yours or whatever?
Yeah. So we have the culture stored away in the original blend that we got from Lars, and every time we grow that, we take it, streak it out of the freezer. And a good microbiologist would start your culture with a single colony off of a plate, but obviously if you do that, you don't have a blend. So for Hornindal, we do it a little differently, and we'll take a wide swath off that plate to make sure we're getting a sampling of everything that we started with, just to maintain to the greatest degree we can the blend as it was when we got it.
How long does it take to grow up like a big, 30-barrel batch, or something of that yeast blend off of a streak?
So starting with a plate, we can have a 30-barrel batch grown up and harvested in 6 days.
Wow.
The magic of exponential growth.
But you're like constantly oxygenating that medium, right? And it's the perfect environment.
Yeah, and just to start small and just gradually, feed it. Just give it more and more food, and and just keep going.
You step it up, basically, just like small amounts of wort, and then add it to like a larger amount of wort, or how's that work?
It's essentially it. You start with 200 milliliters and swizzle some yeast right out of the plate in there and grow that up. Step that up into 1 or 2 liters and just keep that going.
Cool. You mentioned too, and this is something that I'm kind of interested in. I think that at one point in time you had done research, or you have actually produced yeast that you've created via like yeast mating, like you've basically made a different yeast to find. One of them was like a Saison yeast, right?
Yeah. So our Saisonstein's Monster is a hybrid.
Okay.
Yeah, of the DuPont Saison strain and the French Saison.
So it's not a blend of yeast in there. It's a I guess more or less proprietary one that you've created by and tying this to the earlier conversation of proprietary and intellectual property. We literally can patent those things because we created them. There was the hand of man was involved in creating this thing. It would not exist but for our intervention.
So you can patent living things like that you created.
So in that case, ABC Yeast Lab released a new strain, and you somehow got it and tested it and found out it was genetically your strain. Then you can fight that.
Yep. And in that case, it doesn't matter what they call it. They can call it, Saison 5. And they don't have to call it Seisenstein's Monster. We could still go after them.
That's interesting. Oh, that's cool. I didn't actually know that. I thought it was like a blend of yeasts. So for Sundew and Bonanza, then that's the same because.
Yeah, I mean, well, to add, there's always wrinkles. So those are.
Can you explain what those are? Yeah.
Yes. So Sundew and Bonanza are essentially. So Bonanza, for example, is our Hefeweizen strain 021 that we just made it non-phenolic. So the reason yeast in their natural state are phenolic is they have a gene called FDC1, which stands for ferulic decarboxylase. Which takes ferulic acid that's naturally present in wort, and they convert that into 4-vinyl glycol. And 4-vinyl glycol is what we smell, perceive as clove. So any American yeast, lager yeast, English yeast, they've all got a mutation in that gene that basically makes it stop. It doesn't express, so it has a premature stop codon. That's what if you're a biologist. That prevents it from making the whole protein. And so the result is it's non-phenolic. So we basically induced that same change in Hefeweizen and our Belgian A strain. So that it's got now has the same mutation that any American ale yeast or English ale yeast has. But we used, CRISPR, modern genetic editing tool, to make that very, very precise change. So the neat thing, at least what I find neat, but I'm a nerd, is that, you could take Hefeweizen and Bonanza, sequence their entire genome. They are the same throughout the entire genome, except for that one base change. And it profoundly affects the way that yeast tastes, right? The Hefeweizen strain, traditional one, you get the banana and the clove. For the Bonanza, it's just banana because it no longer has the ability to make clove because of that single base pair change. It's just, I think shows how powerful, single gene changes can be. So since we induced that change, in theory, we could patent it, but there's all sorts of other, this is where the crossover with my past life actually comes in handy. It's just, there are other bars to patentability in that case. It's something has to be non-obvious, and there's basically been publications out there where everybody knows that that's why strains are non-phenolic are non-phenolic. So the patent office, if you tried to patent something like that, would say, well, it's obvious, it's known to one of ordinary skill in the art that you could make that change and have a non-phenolic version of the strain. So we're not going to give you a patent for that. So while it's technically patentable, it's we'd be very unlikely to actually have a patent issued on a strain like that.
Wow. So since you're using CRISPR, you are, and I may be overstepping because I'm not a scientist, you are genetically modifying that yeast then, right?
Yeah.
So do you into the GMO regulations and?
So no, we submitted what's called a GRAS notice with the FDA, which means generally recognized as safe, where we basically spell out what we did, how we did it, and say it's safe because it's a change that already exists in other brewing yeast. You can actually use that it's the same route that, say, Lallemand used for Sourvissier, and that's actually a transgenic. So what we did would be considered cisgenic. So we used a change that's already present in that species, in other versions of that same species, and applied it to this particular strain. Whereas something like Sourvissier has a gene that does not exist in any form in Sourvissier, and they took it from another fungus that the LDH gene, the lactate dehydrogenase. that makes lactic acid, and they moved that into Cerevisiae. So that is a transgenic organism, GMO by what we would normally consider GMO. But they're filing through the same pathway. Just basically submit a notice to the FDA that says we think it's safe because like this gene is already present in other food products and things like that. So, in my view, it's unnecessary for us to even do that. But we talked to the FDA and said, this is what we want to do. Do we need to file a GRAS notice? And they said, yes, you should file a GRAS notice. Like, okay. So we did. But it is, in our view, this is GMO light. It's, we are just at this stage, just inducing changes that are already present in other brewing yeasts. It's not transgenic. I have nothing against transgenic, and we'd like to get into those sorts of things as well. But this is as light as it comes with GMO.
All right.
Yeah, that's. I had no idea when like that deep. That's really cool. That's really crazy. Do you have? Well, you probably can't say, but is this something that like you guys are like pretty passionate about right now? Is like the yeast, breeding and gene modification and stuff like that. Like I've been telling people that I feel like there's only so many like yeasts that are going to produce so many flavors, and we've discovered a lot of them, and they've been around for a while. And so what's next is maybe like blends and modifications and stuff like that. And I feel like Kvike obviously was, I don't want to say discovered, but came into popularity quite a bit the last couple of years, and that's redefined a lot of different ways of brewing and stuff like that. And I don't know what is that like a focus for you, or?
We look. They're used for different things. So I view. Hybridization you can actually use kind of like you'd use. You could in theory actually create Bonanza or Sundew by crossing that strain with a non-phenolic parent. A non-phenolic strain, and then it would still be phenolic because it would have one copy of the phenolic gene, the intact gene, and one that's not. So then you'd have to do a long series of back crosses. But ultimately, you could, in theory, cross something enough times to get where 99% of its genetic material comes from the strain you want, but it still retains that non-phenolic version. So you could accomplish these things by breeding too. It just takes, months and months and months. And you're ultimately going to end up with the trait you want, but you're going to have some other parts of the genome sprinkled in there too. So it's not going to be this single change. Whereas you can use a modern genetic tool to it's literally that one single change. It's very, very targeted. But the other thing I think you can do with hybrids is yield things unexpected, because you're mashing the genomes of two different yeasts together, and you might think you might end up with something with these characteristics, but it's really kind of unpredictable. So to me, that's where there's a lot of potential for just new things. You have new performance, new flavor combinations, just because it's so complex. You're mashing together so many different gene types that you just don't know what you're going to get. So there's a lot of potential in that.
You guys have a whole R&D department, like basically that sort of like does that sort of stuff, like on that's like their whole job is to.
Oh yeah. So it was about almost 2 years ago now, we hired Laura Burns, who was the head brewer at Great Central in Chicago, big contract brewer. And she has, like me, a PhD in microbiology studying yeast. So that's her job, is to do. She's the one that formulated proper seltzer and created Bonanza and Sundew. Working on lots of hybrids. We've got that's her full-time job, is playing in the lab and creating strains for us to brew with and sample and all that. So yeah, we're definitely putting resources into that.
That's really cool.
Awesome. So to take this back to the home brewer scale, some of the things, and they may be old wives' tales. I guess that's what I'm trying to clarify is, when I started learning how to brew, and I'd buy my package of yeast and pitch, and I heard some people were washing yeast or reusing yeast, and I've also heard people say, well, they get mutated, and if you brew 2 or 3 batches with them, you're going to get weird flavors. But you're talking about taking a strain and add a flavor to modify a flavor, taking months and months and hundreds of different variations or back crosses on that strain to get a mutation or a modification in place. Do you think from the homebrewer scale that we really are getting mutations in a 5-gallon and 2 to 3 5-gallon batches?
Yes, but I also don't know. So yeah, I mean, every time a yeast cell reproduces, it's going to mutate. I don't know. I should know this, but 1 in every whatever million base pairs it messes up when it's copying. But a lot of those changes it makes are what are called silent mutations. So it doesn't actually change what the protein ultimately looks like, or it happens in a region that's not even coding for a protein. So most of these changes it makes are not noticed. But also just imagine if your non-phenolic yeast, one of them in your culture actually mutated to being phenolic, like that could theoretically happen where that switch happens back the other way. It's gonna happen in 1 cell at most in this culture of millions and millions of yeast. That would never be, you can brew with that for 10 generations and you're not going to notice phenolic because it's not enough of it in that population to ever be noticed.
Right.
I think the more likely explanation for off flavors is that you introduced something else that in 2 or 3 generations in that just eventually got out of hand. I don't think it's not a mutation that's inducing. I mean, it can be an unhealthy culture that is you have autolysis problems going on or something, and like that's the source of some off flavors. But it's more likely contamination, something like that, but not mutation.
Yeah, I can imagine. It's just us homebrewers with lack of equipment or improper cleaning and contamination. I get that, but yeah, it was always like, oh, it's going to mutate, and I was just like, how is that possible?
Yeah, I mean, so the answer is yes, it is mutating, but not in it's unlikely to be in a way that's going to be noticed.
And personally, I'm a big fan of buying a brand new pack of whatever yeast, Omega or whoever. I'm not into washing and cleaning and storing, and that's a pain in the ass. And for you two that like to do that, more power to you.
Yeah, I mean, some people enjoy that aspect of it. I mean, being a home scientist is cool.
Right.
So yeah, it's doable if you have the right tools and know-how, for sure.
So you mentioned earlier in Buzz that you actually had access to a yeast farm or a yeast bank. Was that done? Was that put on by the college or just a guy in your club?
It was a guy in the food science department. I think he was I don't know if he was faculty of some sort or a graduate student or something in the food science department who would kind of do it was just, stored away in a -80 freezer, and you'd say, then they had a list of what they had somewhere. So you'd be like, hey, can I get a starter or whatever? And he'd swizzle it out into a test tube and hand you that test tube. And
so you had access to equipment that homebrewers typically would not.
Yes. The -80 freezer is definitely not something that most people would have at home.
I rock my chest freezer out with a insulated cooler and hope for the best.
Yeah,
I've had luck with a couple-year-old yeast, but
oh sure, yeah. I mean, I think what the -80 gives you is basically if you don't lose power, that's in perpetuity. As long as humans could test, those yeast are going to be alive.
That's crazy.
And in your home fridge, if you have one that's doesn't have a defrost cycle, so it's not constantly thawing and refreezing, I'm sure those would be intact for decades as well. It's just -80 is just the further away you get from any chance of a thaw and a refreeze, that's what's damaging the yeast. The ice crystals, as they're forming, can basically puncture the yeast. And the colder it is, the less likelihood for crystal melting and reformation.
Do you guys use like a glycerin solution like to freeze yours, or what's like the ideal percentage of like glycerin in solution?
15%. So we'd have basically like a 30% glycerol stock solution, and you dilute your culture 1-to-1 to end up with 15% glycerol. But that's at -80. I don't know if that would probably freeze. I mean, basically, I don't know if that would freeze at normal freezer temperatures.
I run like 12.5 usually, by my rough kitchen math, and it freezes pretty good. So yeah. Oh, that's cool. Man, do you still homebrew, Lance?
I haven't brewed myself in years. Certainly when we were starting out, I mean, I was doing all the test batching too, and we didn't obviously have orders with the frequency we do now, so there was a little more time to kind of brew experimentally too. So I did it then. But man, the last time I probably ran a brew myself was a good 4 years ago.
That's not too long.
I mean, we have somebody on staff whose job is actually to homebrew effectively. We have like a 1-barrel Blackman electric system that will brew, do a 6-way split, 6 different yeast strains, and then do all sorts of blind tastings and evaluations. So that's somebody's job, is to run that system.
That's cool. Do you guys like ever like serve any of that? Like, COVID aside, would you have like beer for people to taste like on tap there?
I mean, not like a public thing, but we do. Yeah. I mean, like when customers, I mean, we'd have, especially, our neighbors, Old Irving, we'd have them down. We're like, we got a cool strain that we just made and we brewed with. You want to come try this? So they'd come down and try it. So yeah, we used to do more of that kind of stuff, obviously. Now it's just for internal consumption.
I got a crack in here. But as far as like seltzers go, what's your favorite flavor?
So when in our experimentation, we've done one that was meant to be like a grapefruit Paloma that I really, really liked. So I don't remember. I didn't make it, so I don't know what all was in it. But
I'm ignorant. What is Paloma?
So a grapefruit Paloma would have grapefruit juice and mezcal. So it has a nice, smoky component to it. So he used some sort of tea, I think like a smoked tea, to get that aspect of it. Obviously not spiking it with mezcal. And then, grapefruit rind, I think, and I don't know what else he did with it, but I really liked it. I mean, it was akin to a cocktail. But just the creativity, and this is what I'm getting at with like the opportunities for creativity, when he's using a smoked tea to evoke mezcal. So there's really an opportunity for creativity in this realm.
That's so cool. I'm nowhere near that creative. I don't know. I try like, beers or seltzers or like meat or whatever from like, different places around, even just the state, and just like things blow my mind. Like I had like a Moscow Mule sour the other day. That was just like insane. Or like a Rooibos Blackberry sour IPA. That was like really good, and like it's like that doesn't sound like it should work at all, and I'm not even sure if I should buy this, but
right,
don't know until you try. But I mean, there's some very fruity, fragrant teas that work well in beers.
Yeah,
that's actually how I got involved with this club, and I don't know if David's watching, but I was in an older brew club in our area, which I appreciate Buzz's crazy name. I used to think so. The original club in Kalamazoo is the Kalamazoo Libation Organization of Brewers, and I always thought that was kind of a stretch. And then I hear buzz, and I'm like, wow, you guys, bam, you win.
Really on the nose.
But I was trying to do a beer with Earl Grey tea, and they told me about this other club member that had brewed beer with tea, and I think it was probably the same tea you're talking about. The smoked tea is Lapsang Souchong or whatever. It's nasty, disgusting, horrible shit.
I think that's from Avatar: The Last Airbender.
Yeah, but we're at Bell's, which is local here, and I see this guy and I go up to him and I'm like, hey, you brew beer with tea, and he's like, who the fuck are you? But now we're good friends, and yeah, I've used tea a couple of times in beers to get some crazy flavors, and David's done the same thing. I know he did. What's the pea berry tea? He brewed a lager with pea berries. If he's watching, he's going to yell at me, or when he watches this next week. But that aspect of the tea is it gives off a purple, pink, blue color, right, depending on which way you're looking at it. And his beer was the same way. We were at New Year's last year at his house, like pea blossom. Yeah, if we shine the light this way, it was like purple, and this way it was pink. And
well, and it's pH sensitive too, right? So you get people that make sours with it, where I think it gets like purple, like hardcore purple.
Yep, it's a really cool shit.
Yeah.
It has like green colors and stuff like that too. I think it's some of the higher pHs. Butterfly pea flowers. Thanks, Brandon.
Yeah.
I think he called it Smurfette's lady parts.
Tasty.
He's a creative brewer. So trying to go through. So as a home brewer, Lance, and maybe with the history, and you know how we do things on the home brewer scale, is there any tips or tricks or advice that you would have, or how we handle yeast on our side, or what we can do to be careful with our beers to get the best or the most flavors out of our yeast?
Yeah, I mean, I'm a big advocate of just starters as being one of the best ways to ensure consistent and good beer.
Even with your hydrated packs?
Yeah, I mean, that's what if it was me, that's what I mean, even if I had a week-old pack of my own yeast, I would still do a starter. Just because you want it to be, kicking, like fast, right? I mean, a lot of what brewing is about is overwhelming the possibility of anything else taking over. I mean, you can have really good sanitation and all that, but, just something else might get in there in some small quantity. And so just overwhelming the system with what you want to go in there is going to, most of the time, yield a better product. And really that's the way we work on the pro side. Is there are other companies that make large amounts of yeast, and then they'll divide it up into packages and it sits in their cooler until it happens to be ordered. But on the pro side, we don't start growing it until it's ordered. So you can't get yeast from us to brew a 15-barrel batch tomorrow. But when you get it in a week, it's going to be as fresh as it can possibly be, because that's the way we think you should be brewing. And the way to achieve that on the homebrew scale is to do a starter. And it still kind of blows my mind that all the preparation that goes into brewing at home, I mean, cleaning all the equipment, getting it in shape, having the whole brew day, for a lot of brewers, just making a starter is like a bridge too far. And like, I don't understand it. You're already putting in all this effort to make the beer. How much more effort is it to do a starter? I mean, you can prepare you can sterilize wort with a pressure cooker in advance and just kind of have it ready when you need it. So
buy proper starters.
Yeah, that was going to be next. Yes, you can. We've done that for you and canned it, and again, super easy. It's right there. You just cut it in half with water, and you're off to the races with your starter. It's just, I mean, it proofs your yeast. Like you don't, know necessarily how it got from point A to B to C to D to your house to brew. Maybe UPS lost your the package we sent to the homebrew shop and we didn't know about it. I mean, and so doing the starter is just a way to also proof your yeast, just make sure it's growing.
It's funny. It's the one thing I think that as a home brewer that I've actually moved away from. I started making starters, and as I've grown and improved my beer, and I started actually oxygenating instead of aerating, I stopped creating starters.
Yeah. I mean, oxygenation is another great one. Yeah. I mean, and we've done, you hear all the time, you got to oxygenate or at least aerate, right? Or it's,
Yep.
and I mean, we've done an actual experiment where we, made some wort, and one we oxygenated and the other we didn't do anything to, pitched the same culture, split between those two, and you see a massive difference in attenuation and speed of fermentation. And I mean, it makes a huge difference. Yeast need it. It is an essential. I mean, it's no less essential than the sugar in there. I mean, they need oxygen. It's a component of multiple things in the cell. They need it for the ergosterol that's in their cell walls. They need it for the fatty acids that's in their membranes. I mean, they can't survive without these things. So yeah, oxygenation is definitely high up on the list. The other thing I would have said before Kvike came along was temperature control. So, it still holds true if you're not using Kvike, but good temperature control makes a big difference in beer quality. So, holding your ales at your clean ales at 68 degrees or something, and, keeping your lagers cold, things like that. But, obviously Kvike, if you're using Kvike, there's a little less importance on that.
Mm-hmm.
So those are the big ones: starters, temperature control, sanitation, another big one.
Sanitation is one. It's like no matter what book you read or what person you ask or brewer you ask, usually the answer is sanitation. I was glad to hear you say starters. It's not that I'm against them. I've gotten lazy and I moved away from them. I have all the stuff and I have the time. You're right. I work from home. It's not like I can't, go down today and do a damn starter.
Yeah. We all have to budget our time in different ways. I mean, it's not you're doing something else, and that something else is more valuable, and that's why proper starter exists. I mean, DME is still attainable out there to make your own starters, but we make a ton of proper starter because apparently people like to shave off, half hour, an hour of the process.
I mean, for like $4 whatever it costs. I don't know. My time's worth that, I guess. Yeah.
Yeah, I mean, I mean, we all spend. I mean, it's a hobby, in large part, and dollars we spend on hobby aren't necessarily the smartest. There are other ways to do it, but that's not the fun part of the process for you. So you buy a convenience product for that part of the process because
everybody starts getting into home brand to save money on beer, right? And then
yeah, it's the biggest fallacy.
What happens. Yeah,
right. Yeah. No, I mean, no, it's a fallacy.
It's totally, people that think that I'm going to get into homebrew to save money, they're so wrong. The story I told is like I went into this. I told my wife I'm going to go to the store and spend $100, and I went to the store and spent $600. And 600. Yeah, and then so that first batch of beer was what. $20 a bottle. I mean,
it's gotten cheaper since.
And then I justify that with her and say, well, the more I brew, the cheaper they'll get, and then I can get down to, maybe I can get down to a couple bucks a bottle or whatever. And then I got into fruit beers and holy shit, I mean, now I'm buying 30 pounds of fresh raspberries. That beer is $1 a bottle.
Yeah. But good beer that you made.
That's right. That's right.
I've always told people my philosophy for brewing beer is basically doing the least amount of bad things possible. Don't focus on getting everything right. Focus on doing as the least amount of things wrong as you can. You don't have to aerate, but if you can, that's better. You don't have to make a starter, but if you can, that's better. And just kind of work backwards from there. That's how you make your better beer. And sometimes, equipment and stuff plays a part in that, but the temperature control and stuff like that. But I mean, you can use Kvike yeast to bypass that, to a point. And
and I think part of it too is as your palate gets more sophisticated, you worry more about certain. I mean, oxygen ingress at packaging is, I probably was terrible at this when I first started brewing, but I didn't know what I was, if something happened to be oxidated, or I was brewing styles where oxidation wasn't as big, everybody's making amber ales when I started. And a slight bit of oxidation in an amber ale is probably not going to be noticed, because that's how you can make your IPA taste like an amber ale, by oxidizing it. But if you're brewing a hazy IPA like that, you will notice if that's heavily oxidized, because it looks terrible and it blunts the hop character. So it also depends on what you're brewing and what the sophistication of your palate and all these things.
I don't know if I should even bring this up, because I don't know if you know or want even to answer it. It was either New Glarus or New Belgium Brewing did a study quite a long time ago on adding like a drop of, I don't even know what a drop, some amount of olive oil, to their yeast starters in lieu of oxidation or oxygenation. You have any like thoughts on that? Because like that comes up on like maybe a bi-monthly basis on like some brewing forums, and people are like, oh yeah, I just dip a toothpick in olive oil and don't oxygenate my wort anymore. What are your thoughts on that?
I have lots of thoughts on this topic. It ignores some important yeast biology. So what olive oil can do is supply a source of fatty acids, and yeast need fatty acids, and they need oxygen to make fatty acids. So, but they also need oxygen to make ergosterol, which is a component of the membrane as well. But the olive oil does not help them make ergosterol. So olive oil can supplement fatty acid production, but it cannot supplement ergosterol production. They also need oxygen to make heme. I'm not as sure on the importance of heme in yeast and fermentation. That might be more of a oxidative growth type thing, which is not as relevant to brewing. But ergosterol absolutely is essential to yeast. And it does not, it just doesn't like that. And nobody ever talks about this. So, I mean, you can read through all these things and they're like, well, I had, of course, it's just chock full of anecdotes. Like I used it and it worked great. Like, well, it worked fine for that time. And really it's the, did we lose?
No, he probably had to hit the bathroom or something like that.
Oh, okay.
He'll be back in a sec.
They disappeared. So, I mean, just in your handling of basically, you don't need a whole lot of oxygen to give them the amount of oxygen they need to make their ergosterol. So, my theory is that just with the handling of yeast, they're getting dosed with enough oxygen to kind of give them whatever they need to make the limited amount of ergosterol they need. But my theory is if you were to only use olive oil and like diligently keep oxygen out, you're going to get a serious drop off in performance by the time you get to like generation 3, 4, 5 repitching. Because they can take their ergosterol and actually divide it between, the mother cell and the daughter cell that's budding off. So that daughter will have some, but they're eventually going to run out as they're reproducing and budding off new cells. So I think the olive oil thing would break down if you were to take it out many generations and be really, really good about keeping oxygen out. So it's, no, I mean, I actually have an outline of a write-up where I want to talk about all this, because you're right, you do see it. Hopefully it becomes the definitive, you cannot use olive oil as it's not a replacement for oxygen. Like, again, it's not that it's a bad thing. It can supplement the need for oxygen in making fatty acids, will not replace the need for ergosterol. So, and the problem is, I've looked into this. So you can't, ergosterol is expensive. Like you can buy it from a science supply. So it'd add on some ludicrous amount of cost to a batch if you were to actually directly supplement ergosterol. There's no cheap source of ergosterol. Geraniol is in the biochemical pathway to make ergosterol. And so that my one thought when I was looking through these pathways, I'm like, oh, geraniol's cheap. Like they can, you can buy that from, because it's used, I think, in, essential, like it's aromatic.
Like a terpene or something like that?
Yeah, so it's in soaps and shit, right? Because it smells good and it can be made cheaply. But then when I was reading more about that pathway, it's upstream of the part where oxygen comes into it. So basically, you're converting these things that you get to geraniol, and then it's some step after geraniol that the oxygen is involved in some cyclization step to make ergosterol. So you can't use, in other words, supplying geraniol would not fix the problem of not having oxygen, because you still need oxygen to take one of those later steps. And then when I looked at what was everything after that oxygen step is a complex chemical. So there's no cheap source of it out there. So to truly negate the need for oxygen, you'd have to directly supply ergosterol and fatty acids. Fatty acids are easy. You can get them on your grocery store shelf. But ergosterol, you cannot. So bullshit. It's complete and utter bullshit that you can use olive oil as a replacement for oxygen. It is not true.
Put your paper out. You heard it here first.
Yeah, I mean, this is why I want to put this together, because I'm tired of seeing it.
It's funny. It's bounced around the homebrew clubs for so long. You know, that's replacement. And I had to run and get some more beer. I grabbed some Hot Butcher. You made me think of Chicago beer. So
nice.
We have a buddy that works at Hot Butcher, and so I went and grabbed a couple of cans of Hot Butcher to get into. On that same, and I don't know if it's the same line, but Drew asked earlier a question about sulfur and K-metis bisulfate. Do you, as a scientist or as a chemist, or not a chemist, I'm sorry, a biologist, have you heard of this at all before, that that when you're dealing with sulfur in your beer, your wine, or your meat, or whatever, that you can kind of combat that with K-meta?
That I don't know. I mean, I'm of the mind that it generally does just dissipate. They're very volatile. The sulfur compounds are usually really, really volatile. So given time, they will volatilize. Or you can blow CO2 in it to just kind of encourage that process too. That will help volatilize it.
Well, that was my thought. In the past lives, I know that sulfur off gases just naturally, but I was going to ask that. Like if I just purge with CO2 and like blow with CO2 and then purge and blow with CO2, is that going to do the same thing as just allowing it to have off gas? Yeah,
that should speed up the process some. I think depending on what the sulfur compound is, even copper can help. It's the reason stills are made of copper. They absorb those sulfur components when they're getting volatilized, or it reacts with them. If you've ever distilled something without copper in the system, the distillate is disgustingly sulfury.
Oh, I never thought about why they would be called copper. Oh, that makes so much sense. That's cool.
That's interesting. I mean, I know in Drew's case, he's not going to, purge with CO2 because it's a wine. But the copper thing's an interesting take on it. Yeah, I didn't know that. I actually didn't know either. Like Brian was saying, that was why stills were copper.
Yeah.
Huh.
Interesting.
Yeah, that's really freaking cool. You had mentioned earlier too, you were talking about like yeast generations and budding and stuff like that. Like, is there sort of like a max generational growth that you would try to like limit your yeast to when like trying to like step stuff up? Because like yeast can only bud so many times, right, before they kind of the cells kind of start to get weak. And
well, they can, but I mean, that bud that it's making, the new cell that it makes, can then
just takes over
bud, that many times. So this is probably another thing I should already know, but there's a maximum number of times a yeast cell can bud. I don't remember if it's like 8, 9, 10, something like that. But again, each cell that it's sloughing off can then go and do that same thing 8, 9, 10 times. So, I mean, there are, we've had customers that, brew 25 generations before they get something else. And they said it was still working fine. I just felt like I should probably start over. And I think some strains are better at that than others. Some just are better in storage than others and reuse. So that's another important thing to remember, is that you need to think of every strain as different. They're separated by hundreds of years of evolution. They've got a collection of different mutations. Some last longer in storage than others. Some repitch better than others. They're all different beasts.
Cool. Well, I don't know. I'm about out, Jason. Like, you got that.
Lance,
I do have one more question.
Okay.
Since we're talking yeast, do you have a favorite yeast strain? Period. It can be because you have a favorite beer that you like or whatever. But like, what's your favorite yeast that you guys make right now? If you had to choose one.
Yeah, I mean, if I had a brewery for my clean ale yeast strain, my house ale yeast strain, I would pick our British Ale 1. Just because it's nice and flocculent, it works fast. It's you can get a little bit of character out of it depending on how you ferment. Like ferment a little bit warmer, you get a little more ester quality out of it. It's quite neutral when you ferment on the cooler end of the spectrum. Crops really, really well for reuse. You can make hazy IPAs with it. I think it is actually related to the famous strain that everybody uses for hazy IPAs. So we do have customers that use it for hazy IPAs. It works really, really well in sour wort. So if you're doing sours, it'll ferment out those like a champ too. So it's just a very versatile yeast. So that would be my go-to as a house ale yeast.
I haven't used it yet, so I'm gonna go pick some of that up next week or something. That's cool.
Yeah.
On that, for those of you watching in our area, in the Keeper's Craft area, South County Homebrew Shop, reach out to Dane there. They carry Omega, and they'll get you anything you need in the Omega space. If you're in an area that doesn't have a homebrew shop that sells Omega, you can hit them up on omegayeast.com. I believe is that correct? To
get homebrew packs from us directly?
Yeah. Could you do that?
We don't actually retail direct.
Okay.
But there would be, there are mail order sources out there, and you can find shops on our website.
I know. Okay, then I'll switch to for Michigan people, AIH. Ventures in Homebrewing can do it, or just hit up your local homebrew shop and tell them to order some for you.
Harangue them on our behalf.
That said, Lance, I really appreciate you hanging out with us and talking yeast and going through this stuff. It's been a great show. Gotten a lot of good comments from users on and offline. So thanks for your time. I really appreciate it.
Yeah, I'm always glad to talk yeast. So thanks for having me on. In
the homebrew demo you did last week. There's Dane, the owner of South County Homebrew Shop. In the homebrew meeting you did a couple weeks ago, you said you had something you didn't want to announce yet, and I believe that was Sundew and Bonanza that was coming out. Is there anything else on the horizon that you can talk about?
I think a lot of the exciting things are going to be in the hybrid realm. We've had some that we've already made that we liked a lot, but want to make them a little better. And again, that's the thing. We have these two particular strains that we put together. All three of its children were different. Again, it just depends on which kind of random assortment of genes come together. And all three were really, really good, but I'm looking for something magic out of this one because it's just but I can't really, it's a neat combination. I think it's, I think we're going to find some neat stuff in the hybrid realm.
Awesome. Well, that's it, folks. Hit up Omega, your local homebrew shop, South County for us, for the keepers. And before we go, I want to point out next week, we're going to have two ladies from the AHA that are going to be on to join us. So from the American Homebrewers Association, are going to come on the air and talk.
Two local to Michigan.
Local to Michigan, yep. A lady named Amy from Stormcloud and another lady named Gail, and they are our Michigan AHA representatives. So they'll be on with us next week talking all things AHA. I'll get that post created hopefully tonight, if not tomorrow. And then I think December's got some interesting stuff coming in store, but nothing I can announce yet either. So
great.
So cool. Yeah, man. Try out Omega Yeast. Proper starter, proper seltzer is amazing. Thanks again.
Good.
Easy, everybody.
Thanks, Lance.
Thanks a lot.
Good night, guys.